Applications of agriculture biotechnology. Part 2. Molecular diagnostics. Molecular diagnostics are methods to detect genes or gene products that are very precise and specific. Molecular diagnostics are used in agriculture to more accurately diagnose crop or livestock diseases. Vaccines. Biotechnology-derived vaccines are used in livestock and humans. They may be cheaper, better, and or safer than traditional vaccines. They are also stable at room temperature and do not need refrigerated storage. Biotechnology-derived vaccines are used in livestock and humans, cheaper, better, safer than traditional vaccines. Stable at room temperature, do not need refrigerated storage. Tissue culture. Tissue culture is the regeneration of plants in the laboratory from disease-free plant parts. This technique allows for the reproduction of disease-free planting material for crops. Examples of crops produced using tissue culture include citrus, pineapples, avocados, mangoes, bananas, coffee, and papaya. Flowers. Gene identification and transfer techniques used to improve the color, smell, size, and other features of flowers used to make improvements to other common ornamental plants, in particular shrubs and trees. Some of these changes are similar to those made to crops such as enhancing the cold resistance of a breed of tropical plants so it can be grown in northern gardens. Flowers can be of different size, shape, color and smell. Common flowers are rose, lotus, marigold. Nutrient supplementation. In an effort to improve human health, particularly in underdeveloped countries, scientists are creating generally altered foods that contain nutrients known to help fight disease or malnourishment. An example of this is golden rice, which contains beta-carotene, the precursor for vitamin A production in our bodies. Resistant crops. Pest resistant genetically modified crops, primarily cotton and maize. Genetically modified, so they are toxic to certain insects. They are often called Bt crops. The introduced genes were originally identified in a bacterial species called Bacillus thuringiensis. Tissue culture. Introduction. Tissue culture is the term used for the process of growing cells artificially in the laboratory. Tissue culture involves both plants and animal cells. Tissue culture produces clones in which all product cells have the same genotype unless affected by mutation during culture. Tissue culture. It's not that difficult. So yes, it is visible now. So here is Embryo donor black. ES cell trophoblast. Culture of ES cell. Embryo host. Albino. Micro injection of ES cells into embryo. Inject embryos into uterus of surrogate mother. Chimeric progeny mice. And here we have plant tissue culture. Overview of the tissue culture process. Excised piece of leaf, isolated cells and plant. Then leaf and then plant, small plant and then more plants. Brief history. Tissue culture had its origin at the beginning of the 20th century with the work of Gottlieb Haberland. Plants, Alexis Carrel animals. The first commercial use of plant clonal propagation on artificial media was in the germination and growth of orchid plants in the 1920s. In the 1950s and 60s, there was a great deal of research, but it was only after the development of a reliable artificial media. Moreshige and Spooge and Spooge 1962. That plant tissue culture really took off commercially, and here we have critical requirements. Tissue culture, both plant and animals, has several critical requirements. Appropriate tissue, some tissues culture better than others. A suitable growth medium containing energy sources and inorganic salts to supply cell growth needs. This can be liquid or semi-solid. Aseptic sterile conditions, as microorganisms grow much more quickly than plant and animal tissue and can overrun a culture. Growth regulators in plants, both auxins and cytokinins. In animals, this is not as well defined, and the growth substances are provided in serum for the cell types of interest. Frequent subculturing to ensure adequate nutrition and to avoid the buildup of waste metabolites. Aseptic conditions. Aseptic or sterile or done. Steps of tissue culturing. Selection of explant. Explants are small pieces of plant parts or tissues that are aseptically cut and used to initiate a culture in a nutrient medium. Explants can be taken from different parts of a plant such as shoots, leaves, stems, flowers, roots, and from many types of mature cells, provided they are able to differentiate into totipotent cells. Establishment of the eggplant. Establishment of the eggplant in a culture medium. The medium sustains the plant cells and encourages cell division. It can be solid or liquid. Multiplication. The eggplant gives rise to a callus, a mass of loosely arranged cells which is manipulated by varying sugar concentration and the auxin low cytokinin high ratios to form multiple shoots. The callus may be subdivided a number of times. Dividing shoots, warmth and good light are essential. Root formation. The shoots are transferred to a growth medium with relatively higher auxin cytokinin ratios, promote root formation ready to transfer to soil.